Let's look into these optimal alignment methods. So when we say optimal, that gives us some optimum scores, and we can have some optimal alignment also, but it may not be the exact one. So that can be the best up to that particular scenarios or up to what resources we have. So the objective is finding the path whose total score is maximal, and it will give the best sequence alignments. There are two methods, local alignment and global alignment. We have seen that earlier also. In global alignment, it spans the full extent of input sequences. So actually it covers the entire length of both sequences in simple language. The one most famous global alignment algorithm is called Needleman and Wunsch algorithm. That finds the best global alignment between two sequences. Local alignment is when the alignment covers only parts of the sequences, and the algorithm which is related to that is Smith-Waterman. So Smith-Waterman algorithm finds the best local alignments between two sequences. This approach, in which we are trying to get a optimal alignment, and where we are taking that big problem and breaking that problem into smaller sub problems, we call it as a dynamic programming approach. So this is used to find the optimal alignment of two sequences and the score of that alignment. So this dynamic programming algorithm is the core algorithm which is there in your bioinformatics course, and whenever it's we can say very important in the foundations of bioinformatics, though it's computationally expensive, but this approach is somehow implemented in most of those problem solving solutions. So it's a method in which a larger problem may be solved by first solving to smaller partial versions of it, and in the end we get those solutions and we put them together and our whole problem is then solved. So there are three important steps in this dynamic programming: initialization, matrix filling or scoring, and then traceback. And then the traceback pictures our alignment. So remember, three steps, very important: initialization, matrix fillings and traceback. And these dynamic programming concepts are implemented everywhere, whether you are aligning to proteins, whether you are trying to get the RNA secondary structure, you are aligning two sequences with one another, those DNA sequences with one another. So in any form or any shape, this approach is there. So while we do initialization, what we do over here is we create a matrix with m+1 columns. How many columns we need? One more, since we are trying to compare two sequences over full length or for local regions with one another. So we need an empty additional cell, and we need an empty additional columns. So and same way we need additional rows. Why? Because we want to initialize that. You'll understand when we start filling this matrix. So M and N are basically they correspond to the sizes of those sequences. In the matrix filling, we fill the matrix with highest possible score. We link it to different options and then we pick up the one that gives the highest score. And in traceback, we move from the last corner and follow the arrows and come up, and then our alignment is done. So dynamic programming approach is a very important method, or very important optimal alignment method, and where in this dynamic programming approach we take the big problem and we break it into small sub problems, we solve them, and in the end we integrate our solution.